Assalamu alaikum dear students. Today we are talking about MDK test 2025 biology MCQs part first. Dear students, अभी तक अगर आपने मेरा YouTube चैनल को सब्सक्राइब नहीं किया तो अभी सब्सक्राइब कर लें, लाइक और शेयर ज़रूर कर लें। शुक्रिया। Dear students, MDK test held on 5th October 2025 on Sunday. So best wishes and best luck. Anticodons are present on option A, coding strand of DNA, B, mRNA, C, tRNA, and option D, rRNA. So the correct option is C, tRNA, transfer RNA. Explanation is transfer RNA molecules have 3 nucleotide sequence called anticodon. This anticodon is complementary to the specific codon on the messenger RNA that ensures the correct amino acids is delivered to the ribosome during protein synthesis. Nonsense codons are present on option A, mRNA, B, tRNA, C, rRNA, and option D, none of these. So the correct option is A, mRNA. Explanation is nonsense codons are also known as stop codons, which is UAA, UAG, UGA, are specific 3 nucleotide sequences found on mRNA, messenger RNA, that signal the termination of translation, so that they do not code for any amino acid but instead of a ribosome to release newly synthesized polypeptide chain. Dear students, subscribe now and press the bell icon. Never miss an update from early technologist. So, the melting temperature of DNA is option A. Solid DNA become liquid. B. Liquid DNA evaporates. C. DNA changes from a double helix into supercoiled DNA. And option D. Native double helical DNA is denatured. So the correct option is D. Native double helical DNA is denatured. Explanation is the melting temperature is the key property of DNA. It's the temperature at which the double helix separates into single strands. This process is called denaturation. It is caused by breaking of hydrogen bonds between the complementary base pairs. So this is very important MCQs. The melting temperature of DNA is increased by option A and T content, B, G and C content, C, sugar content, and option D, phosphate content. So the correct option is B, G and C content. Explanation is the stability of DNA double helix is determined by the hydrogen bonds between base pairs. 3 hydrogen bonds which connect GC content, guanine and cytosine, while adenine and thymine are connected together by 2 hydrogen bond. Therefore, DNA has higher proportion of GC pair, which requires more energy and high temperature for denaturation. Mammalian RNA polymerase 1 synthesizes option A, mRNA, B, rRNA, C, tRNA, and option D, hnRNA. So the correct option is B, rRNA. Eukaryotic cells have 3 main types of RNA polymerase, each with specific function. RNA polymerase 1 is the nucleolus and responsible for transcribing the genes for ribosomal RNA, except for the 5S rRNA. Mammalian RNA polymerase 3 synthesizes option A, rRNA, B, mRNA, C, tRNA, and option D, hnRNA. So the correct option is C, tRNA. Explanation is RNA polymerase 3 transcribe genes for tRNA. 5 is rRNA and other small regulatory RNA. These molecules are relatively small and have specific, well-defined functions in the cell. So this is very important MCQs. Mammalian DNA polymerase is in option A, nucleus, B, nucleolus, C, mitochondria, and option D, cytosol. So the correct option is C, mitochondria. DNA polymerase is a specific enzyme responsible for replicating and repairing mitochondrial DNA. So it's crucial for the maintenance of mitochondrial genome, which is separate from nuclear genome, so that the other DNA polymerases are primarily found in the nucleus. DNA dependent RNA polymerase is option A, monomer, B, dimer, C, trimer, and option D, tetramer. So the correct option is D, tetramer. Dear students, if you like this video, please सब्सक्राइब माय यूट्यूब चैनल। अगर आपको ये वीडियो पसंद है, बराए मेहरबानी मेरे यूट्यूब चैनल को सब्सक्राइब, लाइक और शेयर ज़रूर कर लें। शुक्रिया। So this is last MCQs. Non-coding sequences are present in the genes of option A, bacteria, B, viruses, C, eukaryotes, and option D, all of these. So the correct option is C, eukaryotes. Eukaryotic genes are typically composed of coding region, exon, and non-coding region, intron. So the introns are removed during RNA splicing. So this is a distinguishing feature of eukaryotic gene, as prokaryotic genes are generally continuous coding sequences. Thank you for watching my videos.